questions that you'd like to be asked, and what we'll do is we'll then pick out the most popular ones and ask them after the talk. So without any further ado, I'll hand over to Christiane to introduce the speaker.
Yeah, hello everybody, and thanks for joining. So it's really a pleasure to welcome Gérard Lambeau. He is a director at the CNRS in at Sophia Antipolis, that's very close to Nice. And he's a world-leading expert in phospholipases. And initially he started working on venom phospholipases, but he also expanded his work now to work on human phospholipases, which are also involved in autoimmune kidney disease. Gérard started his career in Nice and really never left, which I totally can understand, and we all sympathize. So he studied in Nice and then did his PhD already at Sophia Antipolis CNRS. Had a postdoc there with Michel Lastunsky, and then moved on as chargé de recherche, and now he's director of research at the CNRS. He won a number of awards, including the Translational Research Clinical Award of the Hospital of Nice and 2 Prime d'Excellence Scientifique of the CNRS, a Prix Senior de la Fondation Jean Wald in France, and the French Kidney Foundation Award. And we are very much looking forward to your presentation on phospholipases.
Okay, thank you, Christiane, for the kind introduction. Thank you, Thomas. So I switched to the talk because we have a lot to say, especially me. I'm sharing my screen. Let me know if you have it in a minute. And sorry, I'm just removing this.
Perfect.
Okay, here we go. I think you have the screen on. So thank you again for the introduction. And this is one of the first webinar for me, so I'm not very familiar to do it this way, but let's do it. This is we are moving to the real off-site seminar and probably meeting in a few months. So we need to get experience. It's like 20 years ago when we were moving from the slides to the PowerPoint, you know, we need some adaptation. So today I'm going to, this is our institute here, and this is the sea here. So we have the mountains in the sea in the area of Nice. So I'm going to stretch from venom PLA2 to membranous nephropathy, a rare autoimmune disease. And I'm going to surf on many aspects of this field, trying to give you some ideas on the venom PLA2, on the human PLA2s that looks like the venom PLA2, and up to this podocyte, these cells, which are very, very important for us, because these are the cells that filter our blood, every second, and we don't think about that. But when these podocytes get crazy, we have a kidney disease, and more specifically, we have a glomerulonephritis. And this is bad news for the patient. I will try to illustrate this. So let's move on. So I'm talking to people in the field of biochemistry, and the idea was to show up the details of the PLA2 and the enzymology of these PLA2s. So we are talking about secreted PLA2, which are small enzymes, 14 kilodalton. This is among the smallest enzyme in the world. Think about that, very small enzyme. They are secreted, stable because of disulfide, and they can be present from bacteria to snake venoms and human. They can exist as an active enzyme, but also inactive, and we can ask why do we have this? And in human, they are involved in several diseases. And the bottom line is that these PLA2s can be killing you or protecting you. So this is the yin and yang story about the PLA2s, making them attractive, but also challenging to understand the function. And let's have a tour on the structure of the protein. Is it working? Sorry, yeah, it's working, okay. So this is the PLA2, the calcium, all PLA2s are calcium dependent. And you can see the disulfide, 7 disulfide for a small protein. You can see the active site, and you can see varespladib, the inhibitor, the famous inhibitor I'm going to touch later. And this is it. And what is important to see is that this is a propeptide that needs to be removed, and the PLA2 is in solution, but it has to bind to the lipid bilayer to get active. And this is something very important. And this N-terminus, you don't want it for the PLA2 to bind to the active site. So let's move on. So the first function of this PLA2 is to be an enzyme. And this is not a classical enzyme because it binds to different phospholipids. The substrate is not a single molecule, but it can be very diverse. Very diverse means on cells, on lipoproteins, on bacteria, on viruses, on microparticles released from the cell, etc. And as a result of the enzymatic activity, you will release the so-called lipid mediators. And the PLA2 is one of the enzymes that release omega-3 and omega-6. And everybody knows the impact of omega-3 and 3 and 6, sorry, on cardiovascular disease, for instance. So because of this activity, the PLA2s are involved in multiple functions in any tissues you can dream. The second point is that we found that these SPLA2s, at least some of them, can act not as an enzyme, but as a ligand for different receptors. And the best known receptor we know is PLA2R1. We don't know the signaling. I will comment on this later. And because of this interaction, the receptor is involved in different disease, including cancer, inflammatory diseases, and membranous nephropathy, which I'm going to touch. So this is the outline of the talk. And I started to work on venom PLA2, touching human PLA2s, and with the main idea that we may innovate, we may do innovation by identifying new SPLA2-based therapy. And remember, this is the logo for the pharmacist with a venom to cure disease. So let me start long time ago from venom PLA2 to human. I started to work with venom PLA2, and these PLA2, they are every snake, almost every snake contains PLA2s of different structure, whatever it is. And these PLA2 can be very diverse in a single venom. In one snake venom, you can find up to 15 different PLA2s. And these enzymes can be active or inactive with mutation in the active site, and they can be very toxic, responsible for most of the toxicity of the snake. And they can also have pharmacological properties, meaning that they may cure some disease. So to make a long story short, years ago, we were just making a blast analysis at the time of the Human Genome Project using the sequences of venom PLA2s. And we pick up new genes in that days coding for human or mouse PLA2s. And these are the genes. And we end up with the idea that we have a family of mammalian PLA2s, which are not functional isoforms. We call them PLA2, 2A, 2C, 2E, etc. But just think these are cytokines. Each of these PLA2s are encoded by different genes. And as a result of one gene, of course, the tissue distribution of any of these enzymes in the mouse or any of these enzymes in human, the tissue distribution is just different. These PLA2 is in the pancreas, these PLA2 is in other tissues, and so on. And of course, these PLA2 can be regulated by many conditions, including inflammation. And some of these PLA2 can be down or up-regulated in cancers, etc. So every PLA2 is one gene, one regulation, one function, or more function. And in fact, if you look at the structure of these PLA2s, of course, they are different enzymes. Some of these enzymes, they have a propeptide. Some, they don't have a propeptide. Some have a C-terminal extension for reason we don't really know sometimes. Some PLA2 are crazy, big in size, 95 kilodalton instead of the 14 kilodalton. They are inactive. This PLA2 is in our inner ear. And this is the knockout of this gene in the mouse makes the mouse imbalance. The mouse has problem with equilibrium. Just to give you an example of an inactive PLA2. And then we have these PLA2s, and again, we have an inactive and two active PLA2s. So the structure is different. And let me make just a zoom on the group 10 PLA2, which has a propeptide. And this is the propeptide in the model of the group 10 PLA2. We could not crystallize the pro-PLA2. But the propeptide likely decrease the interaction with the lipid substrate. And as a consequence of that, so we designed protocols to express these PLA2s in a recombinant way in E. coli. And this is a fusion protein for the group 10 PLA2. And this is the simplest production you can do with the methionine and then the major SPLA2. And when we refold the protein, then purify by reverse phase HPLC the proteins, and we measure the enzymatic activity, this is what we see. So if we have the fully mature protein without any additional amino acid, just this sequence, and the mass specs tell us, this enzyme is 100% active. But if we have the same PLA2, but with one methionine at the N-terminus, look at the activity, only 3%. And if we have the pro-PLA2, it's only 0.2%. This is this difference, okay? So it means that a single amino acid at the N-terminus of this PLA2 has a deleterious effect on the enzymatic activity. What you see here is that for another PLA2, the group 5, this is less demanding. The enzyme can afford to have a methionine at the N-terminus. But now, I want to stress the point that if you buy commercially available PLA2, please don't do it. Because this is an example of an enzyme sold by biovendor with an E tag at the N-terminus. Look at the mass spec. This is not very pure. This is not fully folded, in fact. But the enzymatic activity is 0, just because of this extension, okay? And you can buy the other human PLA2s with the same byproducts. And in fact, many companies in the US or probably in UK, you can buy these PLA2s. And I see papers using these PLA2s and saying, we don't see the effect which was originally published by Lambeau or collaborators. And the reason is just because they use stupid enzyme, not active. This is a very important message to the field. So you can ask, why do we have many PLA2s? The answer is very simple, just because there is many lipid substrate. And I remind you that many lipid substrate means what? Means cells, inactivated or activated, making microparticles with different membrane ruffles. This is microvesicle and exosomes. We have lipoprotein in our blood, and these PLA2 hydrolyzed lipoproteins. In the lung, we have lipid lung surfactants. This is a RDS story for the pandemic, the COVID-19 pandemic. We have viruses with lipid, HIV, and we have bacteria, of course. And just think that any of the lipid on this surface might be a substrate for different PLA2s. And this is the challenge. What PLA2 hydrolyze what lipid in what condition? So just to refresh this, the SPLA2 are enzymes with a complex enzymology. The SPLA2 is soluble in the extracellular milieu, and this PLA2 has to bind to the lipid bilayer because the substrate is not monomeric. The substrate is a lipid bilayer. So the PLA2 has to bind. This is called interfacial binding. This is not the Michaelis-Menten reaction. This is the interfacial binding. And this step is key. If the PLA2 doesn't bind, nothing will happen. If the PLA2 binds, it can be with a micromolar affinity for a weak substrate, and it can be with a picomolar affinity if it's a high affinity substrate. And once the PLA2 bind to the bilayer, then start the Michaelis-Menten reaction that you know, one phospholipid gets in the active site, is hydrolyzed, and the product are released. And then the PLA2 can dissociate, or the PLA2 can just move along the membrane to hydrolyze a second lipid. And this process will be very fast, okay? So as the K-off reaction if the K-off reaction is slow, then the PLA2 is very active. If the K-off reaction is very fast, then the PLA2 has a little specific enzymatic activity. So this is the result we have for all the human PLA2s when we test the enzymatic activity on the PG phosphatidylglycerol liposome, a very simple anionic phospholipid liposome. And what you see is that the enzymatic activity is very different among the different PLA2s. This one is inactive because of the inactivation in the active site. This one has 7,000 lower enzymatic activity compared to the 1B. But if you switch to PC, what you see is that the pattern is completely different. This PLA2 is no longer active, and so on for most PLA2s. If you load the PLA2 on the XL, just a live XL at the bottom of the Petri dish, if you add the PLA2s, you would think that there will be a tremendous amount of hydrolysis. In fact, there is no hydrolysis, except for the group 10 PLA2, just because PC is at the surface of the XL, and PC is a bad substrate for most PLA2s. So don't think that any PLA2 has the same enzymatic activity. They have very different enzymatic activity, depending on the substrate you show up. And the same is true on lipoproteins. So this is for the enzymatic activity. Let me move from the receptor story. So long time ago, look at that, I was young, okay? So we extracted from the Australian taipan snake different venom PLA2s. One is OS1, which is not neurotoxic, and the other is OS2, which is very, very neurotoxic. And with these two enzymes as tool, we identified for the first time two receptors, the N and the M type. And the M type is called the PLA2R1 receptor. So we know a lot on PLA2R1, nothing on the N type receptor. We could not clone the N type receptor. So for PLA2R1, it is a C-type lectin member, crazy family. It belongs to, it is very homologous to the macrophage mannose receptor that you may know. And this receptor not only binds the PLA2 in this domain, but it also binds sugars and collagen type 4, for instance. The receptor can exist as a membrane form, but also as a soluble form with a cleavage site here. We are working on that. And now comes the function of this protein. So this is busy. We don't know. We know that when the PLA2 binds to the CTLD5, there's an inhibition of the enzymatic activity. We know that the receptor can internalize and degrade the PLA2. So this is suggesting a role in clearance. On the other hand, some people at least are thinking this receptor is doing some signaling like a black box. And as a consequence of that, papers and papers, including papers with us, has shown that this receptor might play a role in cancer, in cellular senescence. And during the last 10 years, we just identified this receptor as the major autoantigen in this autoimmune disease I'm going to comment, membranous nephropathy. And the receptor is in the kidney in that case. So the receptor belongs to this superfamily of C-type lectin, 300 protein in human. And I just want to stress here that in the snake, but in the serum, there are SPLA2 inhibitors which also have a CTLD domain, just like PLA2R. So this means that from the evolutionary point of view, the snake has selected some PLA2 inhibitor to bind and inhibit the venom PLA2, just like we, with our PLA2R1, we may have selected PLA2R1 to inhibit our PLA2s. And if we expand on that, I don't want to say it, but there is, if you look at the literature, this is a unclear field, but there are different proteins that might interact with venom PLA2s, and also different protein like factor 10A or heparan sulfate proteoglycans or integrins or the VEGF receptor that interact with either mammalian or venom PLA2s. So I told you before there are PLA2 in the venom, But you can ask, now that we know we have human PLA2s, what about the snake? So the snake, we know the venom gland is secreting different venom PLA2s, and this is known for 1 century. But what is not published, or not nicely published, is that in fact, we don't know in the over tissue of the snake, what PLA2 do we have? And thanks to the Genome Project, the genomics and transcriptomics of different snakes, including the python or the king cobra, and there are more genome sequenced nowadays. If you just look at the genes in these genomes, you will see that the 1B, the 2A, or the group 10 PLA2 is expressed in the pancreas, or in the liver, or in the blood, and you can even find PLA2R1, or you can find the 2A in the testes. So the bottom line of this is that the snake, just like us, just like human, the snake have different PLA2s in different tissues. The snake is a smart animal, okay? And the snake is even smarter than us in the sense that the snake has the same PLA2 as us, but in addition to that, the snake has been doing gene duplication, more gene duplication to make new PLA2s with toxic function. And in fact, this mechanism of multiplication of genes coding for venom toxic PLA2s is probably due to a mechanism of accelerated gene duplication, which is called positive Darwinian selection. There are papers on that, I'm not sure you know. So if you open your eyes to the rest of the world, in fact, the SPLA2 are present everywhere in every organism, including plants. There are many PLA2s in plants, also involved in host defense and so on. And if you look at this in a broad way, this is the signature of a PLA2. The active site, very small, and the calcium loop. The rest of the protein can vary in different directions, depending on the species you look at this. Even in viruses like parvovirus, you have one SPLA2 with no disulfide in that case. So the bottom line is that we have different fold for different PLA2s in mammals or invertebrates and so on. This is the plant SPLA2. This is in the worm, and this is in fungi, in truffle. Okay, next time you eat a truffle, think about that. You're eating a PLA2 from the truffle. So the bottom line of this is that the SPLA2 form a kind of superfamily of enzymes, and this is very different from the intracellular PLA2s. They are in our cell. We also have intracellular PLA2s, but they have nothing to do with the secreted PLA2s in terms of structure and the mechanism of enzymatic activity. Even the two types of PLA2 target the same phospholipids. These isoforms have different enzymatic activity or no enzymatic activity, and this is suggesting many things. They can be ligand, and we have this receptor I'm going to discuss a little more. So with this catalog of SPLA2s in human or mouse, or even in venom, and with PLA2R1, what can we do? This was 20 years ago. We were asking after we cloned this gene, okay, so we have to do the inverse genomics to identify the function from the orphan genes we were identifying 20 years ago. And can we go to therapy with that? And I would like to stress that the people doing the job were, of course, not only me, but my colleagues Kei Yamamoto and Makoto Murakami in Japan. And if you Google these guys, you will see the tremendous amount of work done by Murakami in particular. And this is Mike Gelb, the #1 in the world on the enzymology of the PLA2. And what I told you today, I learned from him. So we were looking at the function of these PLA2s in many directions: lipid metabolism, atherosclerosis, immunity, cancer, membranous nephropathy I'm touching, and male fertility I'm not touching today. We had reviews on that, and there's more recent reviews from Murakami. And at the time, by the time we were digging for the function of these PLA2s, there was clinical trials because we were not the only one to work on PLA2s. In fact, Eli Lilly Pharmaceutical and many other companies From the very early days, they were identifying the human group 2A PLA2 as the inflammatory type PLA2, or the arthritic PLA2, or the platelet PLA2, or the non-pancreatic PLA2, etc. Many names for the same enzyme. This enzyme is induced by inflammation. It's induced in any inflammatory disease, acute like sepsis, or chronic like cardiovascular diseases. And the bet was that this PLA2 should be inhibited because this is the PLA2 making inflammation, to make an inhibitor of this enzyme that would do better than aspirin, than paracetamol. That was the idea 30 years ago. So Lilly was taking the lead for about 20 years, making the structure of this PLA2, cloning the enzyme, making the structure, developing the first potent inhibitor, and you see the papers. And there are reviews and reviews on the many inhibitors for PLA2s. They were really drug target by many companies. This is an example from SmithKline Bisham. This is Bristol Myers Squibb. This is from Shionogi in Japan, etc. All the pharmas in the world in the 90s, they were trying to target this PLA2 with different inhibitors. The lead compound, the best compound, was varespladib or indoxam. And you see that this inhibitor can block this PLA2 with an affinity of 9 nanomolar compared to the group 1B, which is not targeted. They did a lot of clinical trial or preclinical trials going to the direction that when we inhibit this PLA2, we block the inflammation. So this was good news, but they went to phase 1 and phase 2 clinical trials with varespladib on sepsis, and the deception was big. The inhibitor fails to improve clinical outcome in sepsis. The inhibitor didn't work in rheumatoid arthritis. The inhibitor didn't work in asthma. And the inhibitor didn't work in atherosclerosis, in cardiovascular disease, with 4 phase 2 and 1 phase 3 with 5,000 people. And not only there was a lack of efficacy, but there was a higher mortality in the patients treated with the inhibitor. Very bad news. We are here in 2040. Sorry, I missed the year. So we are 7 years ago. And at the time this phase 3 publication came out, the company, Antera Pharmaceutical, closed the door the next day. Okay, this was a big deception in the field. And from this was stopping all the research in the private company to work to develop PLA2 inhibitors. You see here that the inhibitor does the job. It inhibits the group 2A PLA2. But what you see here is that the inhibitor blocks many other PLA2s. And this could be one of the reasons it doesn't work. So this inhibitor, in fact, is not a group 2A specific inhibitor as Lilly wanted initially, forgetting about the other PLA2s. It's a pan-SPLA2 inhibitor. And it could be the reason it doesn't work. And then just to show up, because of the ADDovenom consortium, this is a paper from Matt Lewin in the US, repurposing the same molecule varespradib to treat envenomation. So you know in this attendance envenomation, you know this is a neglected disease, and you know there's a need to find new therapies, new anti-venom molecules. And varespladib here is proposed as a new molecule to treat envenomation. And this is Matt Lewin in 2016. And just to make a long story short, I wanted to cite this paper from Nicolas. I'm not sure if Nicolas is attending this seminar. But this paper is impressive because here the paper used 2 small molecule inhibitors. One of them is the varespladib, and this is clearly very efficient in a preclinical model in mice against viper snake bite. So congratulations, Nicolas. This is a very nice paper. If we just look at the literature, it's interesting to see the start, I would say, of a yin-yang story. These 2 papers, they show that when people getting bitten by snakes and undergoing envenomation, In fact, our human PLA2 is increased in the blood because of the inflammation induced by envenomation. And this paper is saying the same thing. So two recent papers, look at the dates, two recent papers showing that our PLA2 respond to the inflammation induced by the venom PLA2. And this paper suggests a mechanistic signaling pathway making the bridge between the venom PLA2 making inflammation and our endogenous PLA2 making signaling around inflammation. So the idea is that the varespladib may treat envenomation by inhibiting not only the venom exogenous PLA2s, but also inhibiting the endogenous mammalian PLA2s. And I just want to point that, in fact, this idea that varespladib or the analog methylendoxam is a pan inhibitor, we published this long before what I said. 15 years ago, we published that the analog of varespladib is inhibiting the venom PLA2s with high affinities just in the same range as the human or as the mouse, sorry, or the human PLA2s I showed you before. So this paper has not been seen, but in fact, we were the first to say that the varespladib and the analog indoxam inhibit venom PLA2s. And I was discussing with Chema Gutierrez in that days, in 2006. He was giving a seminar in our lab, in fact. So there's one more trick. When the inhibitor binds to the PLA2 active site, we have the complex. And of course, there is an inhibition of the enzymatic activity. But at the same time, this complex has a weaker affinity for the receptor. So the inhibitor will block not only the enzymatic activity of the targeted PLA2, but it will also block, in principle, the interaction with a receptor. So the inhibitor can block the biological toxicity because of several mechanisms, not only because of the enzymatic activity. And if we look at the therapy and the fact that the Lilly inhibitor didn't work, my opinion on that is that we need selective inhibitors to target one PLA2 at a time and not to target all the PLA2s together. Just think about cytokines. What would happen if you block all the cytokines together, the anti-inflammatory plus the pro-inflammatory? It will be a big mess. Think about if you block all the proteases in a row, you can expect to have big mistakes and so on. So we need specific inhibitor for PLA2s. So I don't know how much time I have, but I may need 10 minutes if it's okay.
That should be fine. Yeah, that's okay. Okay.
So let me give you two examples. I want to stress this notion of the yin and yang of SPLA2s. And these SPLA2s are important for host defense, but in fact, they are likely important for the control of a good microbiota. And if we expand to the host defense and the subversion of this, we go to sepsis. So we have a kind of range from physiology and a good microbiota to pathology with host defense, with infection, and up to the severe infection, which is sepsis. And you can ask, should we inhibit or promote the SPLA2 action in this mechanism? And back to the sepsis, to the phase 2 clinical trials from Lilly. This is the papers. And in fact, if you look at the abstract, you can read this. The mortality rate was 40% in the treated group, but only 31% in the placebo. Even the p-value is not that strong. But see, when you treat with the inhibitor, you have more death. And the trend of mortality was connected with cardiovascular failure. So this suggests that instead of inhibiting the PLA2 like they did with the inhibitor, we should promote the enzymatic activity of the group 2A PLA2 and maybe of other PLA2s in sepsis. So we don't want to inhibit this PLA2, but we want to somehow stimulate the enzymatic activity of these PLA2s. Let me go through quickly on the host defense, and I just want to summarize what is the good, the bad, and the ugly, the yin and yang story. So some PLA2s even from venom, they can have antibacterial activity. I'm not going to detail this. This is a hundreds paper. Some PLA2 have antiviral activity. I will comment on this. And some PLA2 can be antifungal or anti-malarial, blocking the plasmodium. And we did this in collaboration with people. But the bad news is that the bacteria, they develop mechanism of resistance and even subversion to take the lead, to get resistant to the attack by especially the group 2A PLA2s. We have papers on that in collaboration. And I'm going to discuss this point here on group 2D. And as I said before, some viruses, parvoviruses and CMV, they carry a PLA2 activity, which is important for infection. So in that case, the PLA2 is in the other side. It's not our PLA2. It's a PLA2 that we want to inhibit, the one from the virus. There's no PLA2, as far as I know, in the SARS-CoV-2 virus. So let me, because of the pandemic, I think it's timely to discuss the role of PLA2s in viruses. And long time ago, in '99, we published a paper showing that different venom PLA2 in vitro, this is Typoxine OS2 and the B venom PLA2, this SPLA2 can block the infection, can block, in fact, the entry of the virus. It doesn't block the binding step. It blocks the fusion step. It blocks the step where the virus gets inside the cell to make reverse transcription. We published this 20 years ago. And later on, this paper showed that the human group 10 PLA2, again in vitro, can do the same job. So in principle here, we have PLA2s that protect us against infection. But this is in vitro, and we don't know in vivo. And in vivo, we published the reverse thing. I'm here with Barry Rubin from Toronto. We published that the knockout, the mice, knockout for the group 10 PLA2, they survive better to infection with H1N1, okay? So here, the PLA2 is not good for us. We want to inhibit this PLA2, but we need a specific inhibitor for the group 10 PLA2. And this paper published a year after our paper by Stanley Perlman. You may know this guy, one of the group leaders in the world for SARS-CoV-2 and coronavirus. They published this paper showing that another PLA2, the group 2D, is doing the same thing. The knockout mice for the group 2D survives better to infection to a coronavirus adapted to the mice. So, and we published this year, last year, that another PLA2 is important for allergy, for T-cell allergy for HIV patient. So the field of venom of PLA2 and virus is moving fast. And this is an example of the yin and yang role of the PLA2s from in vitro to in vivo. And again, I wanted to stress that we need specific inhibitors. So the other aspect is that whatever is the role of the PLA2 in infection, in host defense, the SPLA2 can be a clinical biomarker. And we published paper on that, but this is a review, a kind of recent review 4 years ago, showing that monitoring the level of the group 2A PLA2 is a biomarker for sepsis. And I invite you to read this review. And in fact, in collaboration with this company, we developed an ELISA, or we developed a point of care system, and we have more facilities coming just to measure the level of the group 2A PLA2 as a cardiovascular biomarker, but also as a marker in sepsis. So I'm almost done. 5 minutes, it's okay? So this is PLA2R1 and membranous nephropathy. And when we cloned this receptor, it was in the kidney. And I had the chance to come up to this membranous nephropathy by somehow serendipity. I didn't know the function of this protein in the kidney, but nephrologists working on membranous nephropathy, which is a rare human kidney disease, 1,000 cases, new cases in France, the same in UK probably. This is a common cause of nephrotic syndrome. There's no specific treatment except immunosuppressant. The outcome is very variable from one patient to another, so you don't know what's going on for the patient. And this is an autoimmune disease, killing the podocytes I told you before. And for the last 50 years, we didn't know the antigen. We didn't know the molecular cause of the disease. And it turned out that for 70% of the patients, PLA2R1 is the key antigen. And I had the chance to contribute to this paper by helping in the identification of this antigen. And again, this is PLA2R1, which is expressed by the podocytes and is the target of the circulating antibodies present in these patients. And we end up, so I got interested in this disease, and we identify a second protein, fancy name, 25 papers in PubMed at the time we identified this antigen. And this antigen, we published this, is important for about 2 to 5% of the patient. The protein is crazy, another big protein. So with that, we could work with Euroimmun, and this was really impressive. 2 years after we identified PLA2R1, or after we identified THS, Euroimmun was developing ELISA or immunofluorescence tests, which are now used in the clinics every day, everywhere in the world. And this is a paradigm shift in the management of patients and for treatment and therapy. I'm not going to discuss this, no time, sorry. What we are trying to do in our hands, because we are biochemists, and we are doing this since 5 years, we are just trying to identify where on the PLA2R protein, where are the epitopes. And this is a busy thing because this is a structure of a single domain of PLA2R, the CSAR domain. This is the structure of the CTLD1 domain. And this is the cryo-EM structure of the receptor. It's not linear. It's like a snail. And where are the epitopes? This is challenging because all the epitopes in the patients, they are conformational. It means that if you make linear peptide scanning, it's not going to work. This is just a big mess. So we are looking at that. And we have many questions. And about 10 people in my team are working crazy every day to identify what could be the role of this PLA2 in the podocyte, what could be the binding of PLA2 changing the world, the game for the PLA2 antibody, how do we make a soluble protein, etc. Many questions. So what to conclude? 2 minutes. Many things to conclude. And this is the beach on the Promenade des Anglais, okay? So I'm not here, but it might be here. I might be this guy thinking what's next. And to make the big picture, back to the snake. So we have the snake, we have the venom PLA2, and these venom PLA2 exert toxicities like weapons, and they might exert this toxicity either as an enzyme or ligand. And the snake I showed you in the tissue PLA2, they are our PLA2, the same as us, with normal function in host defense, probably function in sperm fertility, function in the cardiovascular system. And these enzymes can be enzyme or ligand. And the PLA2 inhibitor in the snake, what about their selectivity? Are they really selective for the venom PLA2? Or maybe they inhibit also the tissue PLA2. This is a black box. I would love to work in this, but it's impossible to get money to work on that because nobody cares the snake. Everybody cares the human. So these venom PLA2 can kill us, can kill the mice and us, and venomation. But in our body, we have tissue PLA2s. And again, they do normal function, just like copy-pasting the snake. And this PLA2 I showed you might be important as a defensive mechanism for host defense against bacteria, viruses, and so on. And we have PLA2R1 that might be one of the PLA2 receptor inhibitors, just like the snake, that inhibits our PLA2. You can ask whether we have a venom as a human. You know, we are teasing people, telling them, spell your venom, nasty way. So maybe we have a venom PLA2. Actually, in our saliva, we have at least 2 PLA2s. And you know that the venom gland derives from the salivary gland. And in fact, you can expand this to invertebrates. And you have the bee or the scorpion, which are venomous animals. And you have Drosophila and worm, which also have PLA2, but non-venomous. And we expand to the other phyla, like viruses, bacteria, and fungus, and even plants. And this is it. So just to leave you with the complexity of this, these SPLA2 can target cells or can target what I call the non-cellular lipids, lipoproteins, bacteria, viruses, and they can target receptors. And this is busy down here for many functions, which is also very busy. But the bottom line is that from venom PLA2, we can dream therapies. We can dream this PLA2 as therapeutic target, bioactive molecules, or biomarkers of disease. And of course, I didn't do all of this myself. There are many people involved from the lab and from all around the world playing with the PLA2 or playing with PLA2R1. And I thank you for your attention.
Thank you very much, Gérard. This was really informative and I found it extremely interesting. And we have a number of questions on Slido, and please, everybody who has questions, fill them in on Slido. Maybe I can just start reading some of them. So what is known about the inhibitors of venom PLA2 in sera of snakes?
So there are 3 types. This is the publication from many groups. We have maybe one from us. So there are basically 3 types of PLA2 inhibitor. There are 3, but it's easy. Going fast here on one slide. Sorry, it's okay. It's just because it's easier. Oops. One more here. So these are the PLA2 inhibitors. So there are 3 types, alpha, beta, gamma. One is a C-type lectin domain, another is a leucine-rich, and another is a 3-finger protein. So the snake, of course, the snake is playing with very toxic venom PLA2, and it needs highly efficient, with high affinity inhibitors in the blood, because otherwise he will be killed himself, okay? So these are the 3 categories of inhibitor, and the affinity can be picomolar, okay? So we have very high affinity inhibitors. Next, maybe?
Yeah, actually, I would like to add on this one. If it has picomolar affinity, why are people not using these 3 types of inhibitors as in antivenoms?
Yeah, in principle, they can. The problem is the selectivity. So the alpha type is very specific for the PLA2 from the same venom. This inhibitor will not inhibit the other PLA2s. It's not a broad inhibitor. The beta and the gamma, especially the gamma, this is more broad. But the problem is to make the recombinant protein. This could be a job for Renaud, okay, making it in a high throughput way. But I think just because most of these inhibitors were identified in Brazil by people in Brazil, they were really taking the lead of that. And there are reviews suggesting to use this inhibitor even to block the human PLA2s in inflammation. So yes, the idea is very good, and it was in fact the case. But in practice, we need to deal with the specificity of this inhibitor.
Good. So another question is, do the PLA2 cause inflammation, or is this a side effect?
That's a long-lasting question again. So I would say the SPLA2 amplify the inflammation. We have many papers in vitro and in vivo with transgenic mice showing that these PLA2, the group 2A, because most of the work was done with the group 2A PLA2, these PLA2 amplify inflammation locally, for instance, in rheumatoid arthritis. We published this.
So do you want me to ask a few, Christina?
Yes, please.
Yeah. Okay. So there's two actually that are kind of related, and what I was interested in. So you showed some information right at the beginning about how sensitive these proteins' activity is to any kind of modifications in the amino acid sequence. And there's a question here about whether the activity of the secreted PLA2s is kind of regulated in some way, and whether that could be a potential mechanism for that.
I'm not sure I got the point. Can you repeat the question? I'm sorry.
So is the activity of the PLA2s regulated in some way? And I was interested to know whether so you showed that they're very sensitive to kind of changes in the sequence, whether that might be a mechanism for that potentially.
Yeah, so I mean, the first mechanism is the expression level, okay, in different in tissues. So classics, I would say. The second one is the propeptide. So for instance, this PLA2 is secreted as a pro-PLA2 and it's inactive. For instance, in the lung, we could identify the proenzyme circulating, but you need inflammation to get a protease secreted that will activate the group 10. So here you have a protective mechanism to blunt, to inhibit the regulation, okay, to inhibit the active form, the conversion to the active form. And the last regulation is in fact the availability of the substrate. As I said to you, I told you, if you go here, just think about that. If you take a resting cell, a cell not activated, there's no enzymatic activity because the lipid at the surface of the cell is not a substrate. So it's not every phospholipid which is a substrate. You need to show up the substrate, just like the ruffles on the membrane. And this is what we saw in HIV. Okay, when we make the T cells bumpy, as we say, with these ruffles on the cell membrane, you may find the substrate at the tip of the ruffle, while this surface is not a substrate. We also published this for sperm cell. So the PLA2 has a surgery effect. The PLA2 is very demanding on the quality of the lipid bilayer. Okay, that's the point. So the regulation is very fine-tuned at different level. Plus, you may have inhibitors of the PLA2 like you have in the snake. Okay, so this is all the regulation and the complexity.
No, it's really interesting. And I had one other question around, so I was really fascinated, this is good that you have this slide up, by the kind of, these are found everywhere across the tree of life. And I was just wondering if you could comment a little bit more on how else these PLA2s are actually used in other organisms. Like you talked about venom and them being a potent part of that. Are there other areas that they've evolved to be used for other functions?
You mean beyond human?
Exactly, yeah, kind of more broad.
So in plant, for example, so there's not so many people working on plant PLA2s, but in plant, usually the people think that these PLA2 play a role in lipid metabolism, which is kind of obvious. But beside that, some PLA2 from plants, they just have a host defense role. They are important for virus infection in plants and things like that. Then about the mechanism, it's more complicated, but you probably know the lipid mediators, just like prostaglandins, leukotrienes, and you may know the anti-inflammatory lipid mediators, just like resolvins. Do you know resolvins or neuroprotectin? These are based from omega-3. The big guy in this field was Charlie Serran, a friend of mine, okay, in Harvard. Charlie Serran is really the guy at the origin of the concept of resolvin, of anti-inflammatory lipid mediators. And this exists, in fact, also in plant. Okay, so the mechanism can be very copy-paste, I would say, between us and plants. And even in Drosophila, there's one guy, Stephen Stanley, sorry, working in Drosophila and looking at lipid mediators in Drosophila, and I'm discussing by time to time with him. So the mechanism is very general from human to Drosophila and to plants.
Can I jump in with a question again? Because I basically, up to now, I really thought that the the PLA2s are toxic because they have a broad specificity, and when they are in the venom, they just come in and destroy our phospholipid membrane by just chewing it up and releasing all these free fatty acids. But now you are saying that it's not quite this. So what makes the venom PLA2 so venomous and toxic compared to ours? Is it the concentration, or is it really a substrate specificity? What makes them toxic and ours aren't that toxic?
Yeah, so I would say in one snake venom, you can have different PLA2s. As I said, for instance, we have OS1 and OS2 in the Taipan Australian snake. OS1 is absolutely not toxic. It's probably like our pancreatic PLA2. It's a digestive enzyme, okay? So this one, if you inject OS1 to the mice, you will do nothing. But OS2 is very neurotoxic. So just think that in one snake, one venom, you can have a toxic PLA2 plus a non-toxic PLA2. And the mechanism of this, it's the debate for the last now 60 years. Okay, what makes this PLA2 toxic and the other is not? And if I try to summarize in a few sentences, 60 years of science, I'm saying 60 years of science, we end up with the idea that we need the enzymatic activity. Still, we have a paper which is not well cited, I would say, where we killed the active site. We were the only one taking a neurotoxic PLA2, in that case OS2. We published this in Biochemistry in 2006, 15 years ago. So we made an active site mutation on OS2. And then so this enzyme has 500, depending on the substrate you use, because this is the complexity of the enzymology. But this active site mutated enzyme has at least 500 lower enzymatic activity. And now if you look at the neurotoxicity, if you inject the mutant in the brain of mice, directly in the brain, to look at direct neurotoxicity, you lose 15. You have a 15-fold lower killing activity. So you lose 500-fold enzymatic activity, at least in vitro, I'm insisting, but you lose only 15-fold for the neurotoxic effect. What can you do with that? And if you look in this paper, we claim, but I don't have the final proof, to be honest, I'm a scientist. We demonstrate that this mutated PLA2 can still bind to some unknown receptor that we call the N-type, the brain receptor, okay? So the idea here is that the toxicity of a venom PLA2 can be the combination of enzymatic activity plus binding properties, but we don't know much about that. And this is a real debate. And then many papers saying that we need enzymatic activity by different ways they do this. In my opinion, it's not always the best way because they use a stupid inhibitor and so on. And at the same time, they say we need more than the enzymatic activity. But what is more? Is it a receptor? Is it a very specific activity? Is it more a different selectivity for substrate? We call the venom toxic PLA2 the penetrating enzyme. If you look in the very old paper, they call them the penetrant PLA2, which means at the enzymatic level, which means that the PLA2, which means that the PLA2 will penetrate the lipid, will be more aggressive on the lipid, like eating every lipid, making a mess, as you were thinking, okay? So this is possible. So what makes the difference between a toxic and a non-toxic PLA2 might be the different enzymatic specificity for, say, more critical substrate on neurons, the one killing you. So this is a very long debate. I'm sorry, I don't have a clean answer.
Thank you. I think we need to stop, right, Tom?
Yeah.
Yeah, I think we're at the hour, but I thought it was a really fascinating talk, and yeah, it's amazing seeing some of the actual applications of some of this knowledge as well, which is great to see. I think with a lot of these sort of, these areas. So yeah, thank you again, Gerard, for an awesome talk, and thank you everyone for attending, and hope to see you at a seminar again soon.
Yeah, thank you very much for the invitation, and it was a very pleasure. Thank you.